In this video, we're going to talk about introns. Now, when we're looking at eukaryotic genes, most of these genes will have regions that are unexpressed. And primary transcripts are going to be much larger than the actual proteins that they create. So here in this image, we can see a primary transcript, and the dotted line is showing you an idea of how long the mature mRNA will be. And the way that we get rid of these introns is that we have a process that will excise these, and we're going to go over this process today. Then, once we got the introns out of the way, our exons can be spliced together. And in fact, we cut out so much that the pre-mRNA is roughly 4 to 10 times larger than what we eventually end up with. So here is an overall flow of how we make our mature mRNA. So here's our total gene for a protein called albumin. And the capping at the 5' end, as we talked about in the last video, that will happen shortly after initiation. And the splicing out of our introns will happen during elongation. So even when the mRNA is being made, it's going to be spliced. Now, the mature mRNA, we only call it mature after splicing, and it's also been polyadenylated. So once we have our poly A tail in all of our exons, then the mRNA can finally be transported to the cytosol and translated into a protein. So first, let's look at the intron sites, and the splice sites are very highly conserved. So at the 5' site, we have a G and a U almost always. That's what these 100s are telling you. And at the 3' site, you're always going to have an A and a G. So these residues are really defining our junction, our exon-intron junctions for us. So let's take a look at this mechanism to get rid of the intron now. And there's two reactions. The first reaction is that there's going to be a adenosine somewhere, usually 20 to 50 nucleotides ahead of the 3' site. And this is going to form a 2', 5' phosphodiester bond. So we never talked about a 2', 5'. We've always talked about 5 to 3. It's a similar idea, except now the 2' oxygen is attacking the phosphate. So this will go and attack the phosphate of the intron's 5' terminal group. Once that happens, the exon from the 5' end will be released, and you're going to form this lariat structure. Once the 5' end is released, now you have a free 3' hydroxyl. This will go and attach to the phosphate of our second intron, which will break the phosphate bond between the intron and exon. The intron is now fully released, and what's going to happen is that this lariat will break, and then the RNA of the intron will be rapidly degraded. Now this is all done without any energy input, so no ATP is needed for this. And the introns will always be excised in that order, 5' to 3', moving right down the mRNA. So it's not done randomly, it's done in order. And like I said in the previous slide, this is going on while your RNA is still being transcribed. So it's still being created by RNAP2 while being chopped of its introns. And that's it for this video. I will see you in the next video.